And I'm sitting here with my new good friend, Adrian. Adrian, tell him a bit about you.
Well, hi everybody. My name is Adrian Indermaur. I'm from Switzerland and I work for Basel University. And we do baseline research of the Tanganyikan Cichlids radiation, which is one of the beautiful examples of evolution and adaptive radiations that we have in cichlids. Mm-hmm. And we study this radiation since 15 odd years, and our goal is to find out how new species form and what genetic and ecological developments are necessary for this process over evolutionary time.
So I did Tanganyikan Cichlids for 13 years, but my experience with Tanganyikan Cichlids started in like the late 70s. And back then, when you were importing that stuff, there was only one Neolamprologus burchardi. Yeah. And now it's been broken up into a whole group of species and stuff. But I also, like you see in your incredible presentation, you went into you have more tools at university level than we ever had. Like we use basic meuristics and morphometrics and things for basically things that were tangible that we could measure. Yes, now with using DNA and these things, it's changing the whole world. I think in the next coming 20, 25 years, it's going to open up so many things. Like I'm sure you saw that groundbreaking paper by Dr. Melanie Stiassny. Yeah. That changed, that to me, that just shook the world for me. I was getting ready to do a talk somewhere, and it was more of a science-based talk. So I wanted to kind of retrain my brain because I haven't been doing it for so long. So I started looking into cichlids, and I remember just typing a few things on the internet, and I think it was like just a Wikipedia page told me that they weren't perciforms anymore. And I was like, this made no sense. So I started looking more into it. And I was at the Shed Aquarium in Chicago just the other day, and they had Ericatania leucotaniana, the engineer goby. Like, you got to tell me that's the closest relative? It makes no sense now.
Well, that's what genetics at the moment tell us.
Yes.
But of course, genetic methods are developing every time. And I mean, nowadays we can sequence whole genomes and you have all of this information around. Mm-hmm. But the thing is, what is still developing is the way how we deal and process that information.
Yes.
So the genetic information being there doesn't mean that the data that we get out of it is 100% accurate, because the way how we deal with these things is still developing. Yeah. So that's like anything in science, that's at the moment the current state of knowledge or disknowledge, how we like to say it. So that's just at the moment the theory on how these things are genetically related, and especially on a high level, such as like with phylogeny through all fishes, it's very, very difficult to, because they are so distantly related, there's so many genetic changes. You need very good algorithms that calculate these genetic relatedness. Yes. And because of that, there's still a certain degree of uncertainty in there. But I mean, this fish looks so different from a cichlid. But of course, cichlids are more than 40, 60, 80 million years old. Yes. And it's very difficult for us to think in these timelines. And if you imagine the differences in morphology that the cichlids have between each other nowadays, it gets much easier to think about that this might be the closest relative, even though if it's ecologically and morphologically and in any other way very, very different than from what it is.
Yeah, and like we talked about even in my presentation, I showed some examples, like cichlids being a family with 3,000 nominal species now. They all aren't perch-like fish. Like back in the day, you could look at an average cichlid, anything from Central America, a lot of the African stuff, and say, that looks like a perch or a rock bass or a sunfish. It's got that same sort of centrarchid type shape, but that's not what we use in science to describe. And you start looking at cichlids in a whole, just the ones that we know, there's lots of them that are extremely obscure, like the extremely laterally compressed Dimidiachromis from the eye biter from Lake Malawi, or angelfish, or discus. But then you also have the pike cichlids from the former genera Crenocycla. Yeah. Or how about something like Tiliocycla or the Tilogrammas from the Congo? Yeah. Now we're starting to kind of look like Ericatania.
Yeah, absolutely, absolutely. But those are just fish who are ecologically so adapted to a different niche that they had to change their shape. I mean, this Teleogramma, of which, by the way, I just got 6 in my hotel room. I got them to take back home. So these are adapted to highly rheophilic environments, and if you give them enough time, they will adapt their shape to fit exactly in the best possible way their environment. And the thing is, you mentioned before, like back in the 70s, we knew, okay, there was this one Burchardi. the thing back then, of course, we didn't have any genetic information on all of that.
Correct.
And we looked on morphology, but the thing is also, we hadn't looked that well yet. So you say there was this one Brichardi, we just hadn't looked at all the other
and the
variants
exporters,
because people didn't move around that much, and there was not that much effort put into the exploration of the different areas of Lake Tanganyika.
Yeah.
If you imagine Lake Tanganyika has a shoreline of about 1,200 miles, I
exactly,
think.
yeah.
And I mean, that's a large space.
And a
to
lot of the
explore.
exporters then would go and catch
absolutely.
fish that were close.
They were
weren't
just going
traveling
there. They were
hundreds
just
and
going
hundreds
there.
of miles to go and see if something else was there. That was enough.
Absolutely. So now, only in the last 40 years, we have been exploring and uncovering a lot of that diversity that cichlids have that's around.
Mm-hmm.
And in the very beginning, of course, you have an image of a cichlid and it's that it, and then you also have like a search image. That's what I'm looking for. And then you don't think that these weird little things can also be cichlids.
Yep.
But it has been these cichlids who have been adapting to these ecological niches, and that's what for me makes the system such a cool system, because it's this huge diversity and these ridiculous adaptations that these fish show that makes it so interesting. And for us, that's the cool thing, because these extreme cases are like the interesting cases where you go and then look in their genetic code and like try to find the underlying reasons how and why they did all these amazing adaptations.
Yeah. Well, you look at like Lake Malawi, we'll just use as an example. You know, it's a huge lake, almost the same sort of size as Tanganyika, nowhere near as deep and nowhere near as old. But like the entire Mozambique coastline, a lot of it hasn't been explored due to political. And in Tanganyika, you kind of have the same thing with the Congo side. There's a lot of areas that haven't been explored. So there's all sorts of things that, and you know, people got to realize that, you know this and I know this, and some people may as well. But like Lake Tanganyika, these are all rift lakes. So they started as thin chasms. Yeah. And you'll often see in both of those lakes where a fish that'll be on one side of the lake will have a counterpart on the opposite side of the lake that'll be almost identical. May have changed slightly in coloration, but as that lake, as that chasm grew and flooded, you know, these fish have also maybe adapted a little bit different to live on a different niche or a different food source, you know.
Absolutely. One thing I want to mention there is we're thinking of these lakes as like opening up, but you also have to think about the fact that over Lake Tanganyika is 9 million years old at least. Yeah. And over this time, climate in the region has changed a lot. So when the lake has filled up the first time, there was this new lake environment and the cichlids started to adapt to it, and there was new open water and deep water cichlids popping up. But what is also really cool is that through these times, the lake has fluctuated a lot in lake level.
Yeah, you talked about that in your presentation.
Yes, exactly. So what has happened is that this big lake has been repartitioned several times.
There used to be 3 lakes at one point in time. Yes,
and that's how you get these distributions. So if you think like that, for example, Tropheus polii and the nectins are now distributed on the Congolese and the Tanzanian side, but this fish has developed in and they're only in the middle of the lake, so they have, and you go back, and nowadays we can actually phylogenies, we can date them, and we can say more or less when their split was between these two lineages, for example, and then you can compare that to geological data, and you see, okay, this happened in a time point when the central basin was one little lake. So you can imagine in that little lake, the Tropheus polyanectins precursor has developed, and when then the lake fills up, these populations that live around this little lake, then they're squished to each one side, and then you get this distribution that you have now, because they're only on these two sides.
And they can't traverse lake open water because there's other species that have adapted to live
and
there.
now if we look at the distributions of the fish today, you always have to consider that these systems are very, very dynamic over very, very long times. And this all comes together as the distribution we see today, as complex as it may be. But that's the interesting part for me, like to retrace these happenings and how all these populations and distributions came up.
So why Tanganyika? Well, what did you start somewhere else, or but pinpoint why Tanganyika specifically?
So like for me, I have to say it's a chance coincidence, because I studied biology in Basel. And there was a new professor coming there, and he studied Lake Tanganyika. Okay. So in science, it's often that way that research topics are kind of inherited. Because my boss, which is professor of zoology at University of Basel, he was a student of Christian Sturmbauer, which is an Austrian professor who started working on Lake Tanganyika already in the early 90s and late 80s. So this is all inherited down.
Logical progression.
Absolutely,
yeah, continuing
absolutely.
On the work.
But like, why did they start to work on Tanganyika? So these were researchers who were interested in the questions on how does speciation work, and very early on people knew, okay, there's this place, there's these lakes with these endemic radiations in there, and they're very interesting, and they could be a good system for this research. And it happened to be that Christian Sturmbauer and Axel Meyer were these two guys who started to become interested in that lake, and that was just coincided with the time when the first, as you said, when the first fishes came in, like the 70s and 80s. And of course, people got really interested in these fishes, and the same happened on the scientific side. And by then, it's just kind of a chance occurrence. And nowadays, it's basically like that between the different researchers and research groups, it's kind of divided the lakes. So some researchers work on Malawi, some on Tanganyika. And since science as publication is a currency for us, in a way, is also a competitive field.
Yes.
It's like you don't touch each other. You just do that, and we stay in Tanganyika. And it happened to be that we ended up with the coolest lake of them all because it's the oldest lake, it's the most morphologically diverse, which is easy to understand because they had most time to adapt to their ecological needs.
A lot more extremes.
Yeah, because they had more time to change their morphology to an exact ecological niche that makes them deal with it. That's why Lake Malawi fish are really cool, but they're all more similar to each other than the Tanganyikans are. And the extreme case of that is Lake Victoria, which is the youngest radiation.
Right.
And they're
basically all the same sort of shape.
Well, the body shape of them is all basically the same. They started diversifying with their mouths. You had got like neochromis, first algae feeders. You got some predators, but there's not as much variation yet. And that's all of it is interesting in its own respect because these are different levels of these adaptive radiations, and you study them in that respect and in that background.
Yeah, I see. I remember back. I was too young. I was born in the 70s, so in the late 70s was when I started as a kid into Tanganyika, and it was just by chance. And it was Bouchard that started it. But I remember hearing the stories of like in the 60s when the Malawi stuff started coming in, and the blue fish was one fish, and the yellow fish was the other fish, and nobody could breed them because they both had the opposite sexes in different tanks.
Absolutely.
Tanganyika had the same sort of thing with the trophius. Like we didn't know. We were just getting mixed bags all the time, and wild fish were killing each other left and right. It was crazy. Now I'm going to ask you about one specific cichlid. Now, people that have seen me talk, I've talked about this one particular cichlid for a long time. I believe it's the only neolamprologine that's never ever been bred in captivity. And for anybody that's kept these fish, worked with these fish, bred any of these fish, they pretty much all breed exactly the same way. You have some extreme adaptations, like the compressiceps that we'll use to go into a crevice or something like that, but generally they're all pretty much the same. The fish in question is the Neolamprologus sexfasciatus, but the blue form.
Yes.
Everybody can breed the brown form. Everybody can breed Treticephalus, which looks similar, just has one less stripe.
Yeah.
You know, and everyone's bred Julies and all the other Neolamprologines, but like nobody, to my knowledge, has bred this one fish. Now, if we look at the lake, you know, on the eastern coast and the western coast, the yellow form goes down on the southern half of the lake for kind of half the lake. It's distributed very widely on both sides, and they look almost identical on both sides. And the pharyngeal plates look identical for the way that they've evolved, but the blue form lives on the entire bottom point. And like, it makes no sense that nobody can breed this fish.
It's very true. It also doesn't make a lot of sense to me. Like, as I'm also an aquarist, and we have a big fish facility, and I actually kept that fish for many years. They never
13 years for me.
we had him for not that much, but we had him for very long. And I actually tried ridiculous things that I don't like much. I tried like hormone treatments to get them to breathe and everything. But we ourselves, we never managed. I think there are some people in Europe who claim that they made it, but I never seen any proof of that. And it's very interesting. And so for me, the yellow brownish forms and the blue forms that we have in the south, they're actually most probably the same species, of course, with some geographical color variation. And it's actually an interesting case because we think that this color variation stems from the fact that, so in the north of the lake, you have the Tretocephalus, as you mentioned, and in the south, we have the Sexfasciatus. And I don't know if people know that, but throughout Lake Tanganyika, being a black and white striped fish is actually quite rare. There's only 3 to 4 species in the whole lake who look like that. And and their niche is often very particular. They have mulliform jaws. They're actually snail crushers, most of them. And in certain areas, they're quite abundant. And one lives in the north and one in the south. And of course, there are areas where these two species overlap. And in evolutionary research, there's a principle that we call character displacement. So why are these cichlids not all one species again? Because many of them can technically interbreed. And when they live in areas and they look very similar, the likelihood for them to interbreed is very high. And so the idea basically is that in areas where the species are overlapping, they have to maximize their difference, not to be interbreeding.
To maintain the species.
Absolutely. Yes. And that's at the moment one of the theories that could be a reason for why these fish on the west and eastern coast of the southern basin towards the northern distribution overlapping with Trettocephalus are more yellow, because they actually look quite different. Yes. Because the Trettocephalus is completely white and black, sometimes some bluish.
Different dentition too, they're specialized differently.
Absolutely, absolutely. So at the moment the theory why this yellow form is different like that is this character displacement thing, that they have to be as different. I mean, we have the same thing in humans. Why are two neighboring cities always nagging each other? Because they have to emphasize their difference, no?
Yep.
In a way.
All the football teams and sport teams and stuff.
And in the south, the blue population, that's not necessary, so they can look as similar to the Trettocephalus as they want. And it seems actually that by natural selection, this black and white stripe coloration seems to have some benefit, which I'm not completely sure on what that is. It might be some species recognition or not.
Okay.
Now, that was a long story, and I'm coming back to your question, like, why can the yellow form be bred and the blue form cannot? I have to admit, I don't really have an answer to this, but I have a few ideas. And so Lamprologines, I don't know if you know this, but like the Neolamprix buscheri is a deep water living species in Lake Tanganyika, which was brought home by Heinz Büscher back in the 80s, the first time.
It was a hot new fish when it came out. Same with Cylindricus. When that hit, Cylindricus was the same.
And I happen to know Heinz very well, and he told me the story. So he brought home 3 of those. And
Three. That's it.
A male
Just
and
3
2 females. Back in 82 or
Could
something.
have maybe brought back 5 or 6 maybe.
Back in the day, they were diving with it was a different time back then,
and fish had to go into like a pressurized thing halfway up and stay there.
And this fish only lives like below 60 feet or something, or 20 meters. So it was at the time not an easy thing to do. And Heinz brought them back, and he tried to breed them. And he actually, with that fish, had the same problem as with Six fasciatus. And he had this trio back at home, and they would just never breed. And he's actually an old chemist. And what he started doing is, from old papers, he got lists of how the composition of Tanganyika water is with all the minerals. He actually went to the lab, and he got like RO water, and he started
making
punching together his
that's
own
what I do.
That's his own Tanganyika water.
Yeah. Before the internet, I would apply to these countries to get like rain patterns and get all that information that we could. It had to be mailed to you.
Yeah. No, that's how it went, and he actually succeeded on breeding this fish. And then later he gave this trio to Wolfgang Steg, who then described it after him in his honors. But that was a big success for him. And I think it might be something like that. I mean, that logically doesn't explain why this one population in the south wouldn't be able to breed. And we actually, with the Six fasciatus, when I had them at home, we tried similar things. Because
with the water chemistry.
Yeah, with the water chemistry, because the most the crassest difference between Tanganyika water and the waters we have here in the northern hemisphere is that it's more magnesium based than it's calcium based. So we tried doing these things and nothing ever worked. So this day,
Ad Koning's mentioned to me, like I've been talking about this since then, 13 years, and that's the mid 80s, early 90s, and
absolutely.
And I still remember talking about it. Like I was positive it had to be some sort of animal, an insect that would seasonally use the lake, and then that would cause a chemical reaction in the female to produce some roe or something. But then maybe it be strictly like a molluscivore, maybe it's a snail. There has to be some sort of chemical reaction. We can't access that, but that same snail, the female with the gold form, would have access to, right?
Yeah, it's a good idea. But then, as you say, the these areas of the golden and the blue form, they're actually not that far apart. It's about it's about 80 to 120 miles maybe between certain populations of them, and many of the invertebrates, which would be the food items for these fish, don't differ along this range. So
probably not the whole lake, really.
Probably not a lot. I mean, it's one body of water. It's very difficult to imagine that anything would change. I mean, there are certain parameters like biological and non-biological ones that differ between the northern and the southern half of the lake. So there's, like, if you do, for example, stable isotope assessments, you always have to use baseline from at least the sub-basin where you're in, because there are certain chemical differences.
Okay.
But for me, it's relatively hard to imagine that a thing like this would be the cause for such a difference in breeding success. But in the end, I think it has to be something like that there is minor chemical differences around that make this, because there's another level that you could imagine which would differ these species is that there's any differences in ecology or habitat between them. That, like, say, for example, that one species would only breed very deep or in caves, or would breed in a certain
Lad says that the species breeds year-round. Like, I used to think that, I thought maybe it was a kind of a cyclic thing, that it needed some sort of chemical trigger to make it to breed.
No,
but he breeds year-round.
I mean, most of the species in the lake actually have very little year-round seasonality.
Correct.
So there's many species who have like a monthly seasonality because they breed with moon cycles and things like that. And they do that for, like, to avoid having the fry when it's full moon, because then there's a lot of light at night and they're more prone to be eaten by predators. So they're more protected when they're in a certain free swimming stage at new moon. And I think the Japanese have done a lot of work on that, and I think that makes a lot of sense. And you do see a lot of monthly cyclical time intervals in many of the cichlids, but I think that for the Cephasiatus they cannot be, because you would have the same for the both populations. So my suspicion is that it has to be some minor chemical. So there has to be some underlying non-biological or biological factor that does this change. Or maybe they're just, I don't know, maybe it's something from the fish, something intrinsic from the fish itself. But
these are all pieces of the puzzle that we've got to start looking at. That's part of the fun.
And that's I wanted to just say the same thing. I think that's the big fun about it, because also things like that make you think about reasons why things are how they are with these fishes, why they do the things that they do in the way that they do. And once you have a problem like this entices you into going in and disentangling all the threads of this big puzzle. And I think that what you pointed out is a case where we are at the moment not there yet on knowing it.
But that's one of those things that makes cichlids so exciting. There's cichlids for absolutely everybody, big, small, and there's a perfect example of a cichlid that's never been bred by anybody in the world.
I haven't heard about it.
Yeah. Thank you, my friend.
Pleasure.
Absolutely incredible. Where can they check you out? Do you have a website, or how do they follow all this incredible work that you do?
Well, we have a webpage from our research group. It's called SalzburgerLab.org. And this is the homepage of our research group. And we actually have all of our publications are free to download there, because, of course, you know that scientific publications are often behind paywalls and stuff, which makes it difficult for the hobbyist. And we don't really like that, but there's few ways around that. So we have all our PDFs on our homepage.
Well, we were talking about that at one of the meals one day, that that's the real thing that's changed in the scientific community. I know we're getting a little bit off topic, but like in Europe, a lot of the scientific community has embraced the hobby community, because the hobbyists are keeping fish. They're not observing jars. They're not, they're actually physically keeping the fish long term. They can offer things that you guys may never see, like the behavioral traits and things like that in a community aquarium.
And that's actually exactly the reason why I'm here at the ACA meeting, because me as a scientist and as a hobbyist at the same time, I really like to live in this intersecting area between science and hobbyists, and I think it's a very fruitful thing that we can both learn from each other.
Yeah, we're only going to benefit.
Absolutely,
so,
absolutely.
There you have it, my friends. He's an absolute legend. He's one of the most in-demand speakers, and if you guys were here at the convention, you'd understand why. So thank you again, my friend. Take care, guys. Cheers.
Thanks.